Welcome to my second video on the nervous system. This video is going to talk about neurons. Those are excitable cells. In a future video, we'll talk about glial cells. So you'll be able to identify the different types of neurons, their location and function. This is usually an essay question where you have to draw them out, and identify the parts of a neuron and give the function. So, when you look at nerve histology, there are two types of cells actually. There are neurons. These are nerve cells or nerve fibers. So this thing right here is a neuron. It is a nerve cell. It's an excitable cell that can create and transmit signals. Then there's all these other cells around it. This oligodendrocyte, this astrocyte, this I think that's supposed to be a microglial. Oh, that's another astrocyte. Those are glial cells. They're non-excitable cells, meaning they don't send signals. They support, protect, and nourish the neurons. They used to think it was just glue that held it together, so the brains we have in lab, the real ones, are still brain-shaped because of the glial cells. So, neurons. They conduct nerve impulses from one part of the body to another part. So, they have these features. High metabolic rate. They need a lot of oxygen. Long-lived. They used to think you're born with all the neurons you'd have, or you had all the neurons you'd have when you're 2. Turns out they can't be replaced. They can, C-A-N. But they're generally non-meiotic. They don't really want to divide on their own. You actually get new neurons from something called a nerve crest cell. So, let's look at the parts of a neuron. So, let's look at what the parts of a neuron are. So to begin with, we have the cell body of the neuron. So neuron, cell, body. Most neurons are going to have one long axon, A-X-O-N. And these send signals. So the axon send signals from the cell body. Let's put a little nucleus there. Let's put some ER going off. We can't forget the Golgi complex, the pout-pout fish. So, we have the nerve cell body. We have our axon. And there's usually one axon that sends signals. Then, there's multiple dendrites. And the dendrites receive signals. So if we look at this picture, you have multiple dendrites here, and they receive signals. You have the cell body. That is the cell body of the neuron. And then you have one long axon. Now this axon is branched into these things called collaterals and telodendria, which we'll get to in a minute. Let's first look at the dendrites. So the dendrites are these multi-branch things, and they receive signals. They're receiving input from other parts of the brain. Now this dendrite might receive a signal going, you know what, we miss our ex, it's 2 a.m. in the morning, let's text them. Fortunately, this dendrite, and this dendrite are receiving signals that go, you know what, bad idea. So who makes up the mind who to listen to? Well, that is the cell body. So the cell body adds up signals. So it's going to add up all these signals. So this is +1, -3. Our input is -2. Are you going to text your ex at 2 in the morning? I hope not. Now, let's say you've been drinking. Suddenly, these signals are all positive. So you have 1 negative signal, 3 positive signals. So it's 3 plus, 1 minus. That is +2. The cell body adds it up then, and it's like, oh, it's a good idea to text our ex. So the cell body is adding up the signals. Then it's going to send the signal on the axon. saying, okay, let's go to our fingers and type in our X's number, even though we should have deleted it from our phone. So, dendrites are these short processes right here, and they're very branched. Axons can branch. You might have something called a collateral here. You might have something called a telodendria at the end, but generally they're unbranched. They receive impulses and carry them to the cell body. The cell body is called the soma. It contains organelles. So remember this slide from the second lab? Those are all ribosomes. So you're actually seeing the ribosomes. That is why they're little speckles. Now it has a nucleus, mitochondria, free ribosomes, rough ER. Because there's so many ribosomes, this dude named Nissl named them Nissl bodies. No ego there at all. And in an unstained nerve, they are gray. Wait, where have I heard gray before? I know. Gray matter. Make sure you put 2 T's in matter. Gray, they're gray in color. They're responsible for gray matter. So if you hear somebody talking about gray matter, it's cell bodies. Now the cell bodies add up signals from the dendrites. So again, my example, this one said, don't text your ex at 2 in the morning. This one said don't. This one said do. Fortunately, all the negative signals tell you not to. However, after 1 shot of tequila, 2 shots of tequila, 3 shots, and before the floor, you got all these other signals. Uh-oh. Now there's more pluses. You're going to do that texting. Now, this little region of the axon right here is called the axon hillock. What's going to happen is all of these signals get added up, and it's going to initiate something called an action potential. That is going to send an electrical current down the axon. In physiology, you'll probably record action potentials on axons, and they are initiated from the axon hillock. So going into physiology, you need to know signals come in from the dendrites, get added up in the cell body, and then the axon hillock creates the action potential. So it's the trigger zone. So let's talk about axons now. Let's go back to our sketch pad. And again, we have our cell body. We have these branched dendrites. What do the branched dendrites do? That's right, they receive signals. We have our nucleus. We have our Golgi complex. We have our mitochondria. And we have our 1 axon. Now the dendrites are highly branched, and they're very short. So dendrites short, branched. Now the axons might be branched, and they could have a branch very close to the hillock. Now most axons are actually myelinated. So I am going to actually highlight this. And so they're myelinated. So they have this stuff called myelin. And these branches are myelinated too. So this blue stuff is something called myelin. It insulates the neuron. And some of these big branches very close to the hillock are myelinated. These are called axon collaterals, laterals. They are myelinated branches. So branches with myelin. And I'll spell myelin on the slide in a second. Now, at the very end, you are going to have, can you see that? No. At the very end, you have these very small branches. These are telodendrites. dendria. Now an axon, teledendria, is at the end, and it branches to all the effectors where it's going. So remember the neuromuscular junction of skeletal muscle had teledendria. So going back here, we have our one axon, which is a long process, and it's going to multiple places and transmitting information. But there's multiple structures associated with it. So the axon collateral is myelinated. So see these things? Those are Schwann cells. They cover the axon in something called myelin. This is insulating it. So they're myelinated, and usually they're close to the cell body. This one's at the end. Teledendria are unmyelinated, so see, no Schwann cells on those. There's splits at the end that go to the individual muscle fibers or individual glands, and then we have these axon terminals. Sometimes they're called synaptic knobs. You should already know that from the muscle chapter. So we have our neuron. So here's our neuron. What are these branches called? Correct, dendrites. We have our one long axon. Then we have our teledendria and our synaptic knob. Now this thing is going to be attached to the axon of another, or the dendrite, neuron, or a muscle. So the signal must go from here, from the axon, the knob of that one, to the dendrite of this one. So the axon here, signal is going down. There's all these neurotransmitters, and they get released, and they go to the dendrite of the next one. Now there's a gap right there. This gap is called a synapse. So I have 2 minutes now to talk about the 3 structural types. Usually I like asking this as an essay question where you draw them out. For an online format, you might have to just explain them. So there's 3 kinds: pseudo-unipolar, bipolar, and multipolar. This guy right here is a pseudo-unipolar neuron, and it basically has dendrites. So dendrites attached directly to the axon. So the dendrites are directly attached to the axon. So if I do it here, those are the dendrites. They are attached directly to the axon. And these are always afferent. And the cell body doesn't do much, really. So there's the cell body. It's pushed off onto the side. So una means 1. Bipolar, what does bi mean? 2. It has 1 dendrite, 1 axon. So here you have 1 dendrite right here, going to the cell body, going to 1 axon. These guys actually develop from the bipolar neurons. Then what does multi mean? So una is 1. We have 1 process. Bi is 2. Yeah, anatomists got lazy. They're up to 4. They don't want to count anymore, so we have multipolar. So the pseudo-unipolar neurons, only 1 process. Axon is divided into peripheral and central ends, and they tend to be clumped into these ganglions. Ganglions are cell bodies outside the CNS. They're always afferent. Bipolar, bi means 2. It is 1 dendrite, 1 axon. They're on opposite pores. They're rare in humans. They're the rarest ones. They're in the nose and the eye only. Most neurons are multipolar. Many dendrites. So many dendrites. Usually 1 axon. All motor neurons, all integration neurons. So they're actually the most common type.